First off, let's talk about the central dogma, which is the process of RNA aiding in the transport of the DNA's information to the ribosome so that it could produce protein. First step of the central dogma is translation. Here we can see the amino acids. We see guanine, cytosine, thymine, and adenine. And we can also see the hydrogen bonds and the sugar strands that make up the double helix of DNA. So what we're about to do right now is take the RNA polymer that cuts up the DNA at the start sequence, which is called initiation, going by the 5 to 3 preference. And it's going to cut it in half, the certain part from the start to stop. So that's going to be initiation and termination. And as it's cutting it, what the polymer helps do is keep the DNA from unwinding and keeping it in a helix. Now see mRNA sliding into the DNA, which is called elongation. And as it's sliding into the DNA, it's replicating the codons that the DNA has. And instead of using thymine, it uses uracil. And as it's going through the DNA, the polymer is is going ahead of it, is going behind it, helping close up and wind up the DNA. And once the mRNA is outside the DNA, is now called a pre-nRNA. Now, as the pre-mRNA starts leaving the nucleus, it starts getting attachments added onto it. So let's make the pre-mRNA orange, and as it's exporting the codons, it gains a guanine cap and a poly A tail to help it to protect it from enzymes that will probably degrade it or help in the destroying of the mRNA, and it also helps to get out of the nucleus and attach to the ribosome. So now let's move on to splicing. So what pre-mRNA has are introns and extrons. Extrons being red are the usable codons, and introns being orange are the unusable codons. Because of this splicing occurs, so what starts off splicing is SNRPs, or the small nuclear ribonucleoprotein, which binds with other proteins to get rid of all the introns. All the introns, so that only extrons remain. So there's only the usable codons. Now, in the ribosome, we have a new RNA called tRNA, and what tRNA does is that with the help of aminosyl tRNA synthesis, it helps binds tRNA to specific amino acids. So what tRNA does is that it reads the codons, and as it reads the codons, it binds to their specific amino acids, and what it continues doing is making a peptide bond. And once they finish the peptide bond, which is elongation, they release it and free the polypeptide.